Hello everyone. Today I'm going to discuss regarding translation process. Previously we know that transcription is formation of RNA by using DNA. So double stranded DNA was used for the preparation of RNA. Now second important process is translation. Translation process is being done for the formation of proteins by using mRNA, which is single stranded in nature. So these are the two important processes in any organism, that is DNA is used for the formation of single-stranded RNA. That process is called as transcription. This is transcription. For every reaction, we need so many enzymes so as to complete this process at little bit faster extent. The process of formation of proteins from RNA is called as translation. Fine. So now what translation process, how it proceeds, what is being needed for translation process, that we are going to discuss now. See, first of all, let's first talk about prokaryotic and eukaryotic organism. Say for example, you have prokaryotic cell and this is eukaryotic cell. This one is prokaryotic cell and eukaryotic cell. So what happens? We know that prokaryotic organisms do not have well-defined nucleus. So they have genetic material which is present in a cytoplasm only, right? So this is DNA in case of prokaryotes, which is freely present in the cytoplasm. In cytoplasm, this DNA is being used for the production of, you can say, single-stranded RNA. So this is our RNA. Same cytoplasm, in the same cytoplasm, simultaneously, you can say same medium is there. In cytoplasm only DNA is used for the formation of RNA. Now RNA is being used for the formation of proteins. So this helps in the formation of proteins in the same medium, right? So in prokaryotic organism, like a process is known to be like transcription and translations are known to be coupled in nature. Why coupled? See, here transcription is happening, right? Here transcription is happening, and this after that translation process will get completed. So in the same cytoplasm, transcription and translation process both are happening. Now what happens in prokaryotic organism, the time when this DNA is being used for the formation of single stranded RNA, that means when transcription process is happening. When transcription process is happening, not yet completed. Suppose, for example, in case of prokaryotic organisms, transcription is not yet completed. There is a possibility, or mostly it happens, that this RNA which is being formed, till now it is not completely formed. It is under process of formation. And simultaneously, it is helping in the formation of proteins also. That means here, translation process is also happening. That is possible only when there is no nucleus, only cytoplasm is there. That means both the processes are happening in cytoplasm, right? So whenever transcription and translation can happen together, this process is known to be coupled process. That means transcription and translation, both processes are coupled in nature. Correct? That means we are assuming now what? That transcription is not yet completed, and by the time transcription is happening, simultaneously translation also started. That is possible only in case of prokaryotic organism. Why not in eukaryotic organisms? See, in eukaryotic organisms, we have this nucleus. In nucleus, our DNA is present. Now, this DNA is used for the formation of RNA inside nucleus only. So here transcription process is happening. Here transcription process is happening inside the nucleus. For translation process, RNA need to come out from the nucleus. Right? So once this RNA got formed through the nuclear pore, nucleus is having nuclear pore, through these nuclear pores, this RNA will come out in the cytoplasm. If you remember that in case of eukaryotic organism, a little bit further processing was also needed, like capping, splicing, and tailing. Why? Because RNA is formed in the nucleus. Now it is coming out in the cytoplasm for the formation of proteins. So it might not get, you can say, degenerated by other enzymes. Hence, capping, tailing, and splicing process got completed. So now this RNA came out from the nucleus through nuclear pore. Right. Now here, this RNA will be used for the formation of proteins. So protein formation is happening here in cytoplasm. That means translation process is happening in cytoplasm, whereas transcription was completed in nucleus. So this is what basic difference is there in prokaryotes and eukaryotes, that in prokaryotic organism process is coupled. That means transcription, translation both are happening in the same medium. Whereas in eukaryotic organism, DNA is present inside the nucleus. So transcription get completed inside nucleus only. Once mature RNA got formed inside nucleus, it will take an exit from the nucleus, will come in the cytoplasm and will help in the formation of proteins in cytoplasm. So in case of eukaryotic organism, the process is the process is known to be non-coupled in nature. Got it clear with coupled and non-coupled process? In prokaryotic organism, process is coupled in nature, whereas in eukaryotic organism, process is non-coupled in nature. Right? Now, through transcription process, first let's understand what we need, what different kinds of things are needed for translation process. We got this RNA, right, through transcription process. You remember that transcript, this mature RNA which we got has always direction from 5' to 3'. See, DNA was double stranded in nature. The one strand was from 5' to 3', another was 3' to 5'. This was reverse in position. So, RNA can be read from 5' to 3' direction only. Now, we have this RNA. See, you know that for translation process, we need start and stop codon. Now, say for example, the position of the start codon is this one, right? Start sequence is AUG, correct? This AUG codes for amino acid called as methionine. Whereas stop codon can be, we have three possibilities, right? UAA, UAG, UGA. Say for example, this is UGA. Now randomly I have taken one position that start codon is present over here, stop codon is present over here. You know now RNA is too much coiled in nature like this, right? So now we don't know where these start and stop codons are situated. Correct. The size of the mRNA will be too large. So we are drawing certain, you can say length of this RNA. Say for example, start codons are situated over here. Now, what we needed? We need proteins. For protein formation, we need ribosome. So ribosomes will come and bind on this mRNA. So this mRNA will be used by ribosomes. But ribosomes do not know where is start codon, right? How do we start? What is the signal who tells ribosome that now start codon is about to come? Now you have to start the process. So first, let's understand this basic thing, that before start codon, see these are the sequences. Of course, here will be some sequences which will be present, which are not helping in the formation of proteins. Why? Because since start codon is present over here, so protein synthesis will happen from here till here, right? So what about the sequences which are present before start codon and which are present after stop codon, right? They won't get translated, right? So these sequences which are present after stop codon, these sequences are called as untranslated regions. Untranslated regions. In short, we can call it as UTR. Similarly, here also UTR are present, untranslated region. Because protein synthesis need start and stop codon. Before that also, I have drawn small part, right? It can be little bit large also. Whatever sequences are there, those won't help in the formation of protein. So those will be called as untranslated region. But what happens, you know, how ribosome recognizes the position. Say for example, just 8 bases before start codon. Remember, 8 bases before start codon. 8 bases before start codon, there are sequences. which are present. Those sequences are called as SD sequences. Now, what are these SD sequences? These SD sequences are nothing but called as Shine Delgado sequences. Say, for example, here start codon is there. Before start codon, like if you go back here, leave 8 bases. Before 8 bases, here it will be Shine-Delgado sequences. Where? Here. 8 bases before start codon. But that condition is there in case of prokaryotic organism. That means these Shine-Delgado sequences are present in case of prokaryotes, right? So we are just discussing some sequences, how ribosome is getting signal, how it is starting protein synthesis, right? So there should be some spark. That spark that now you have to work, now you have to start protein synthesis. Now start codon is about to come, right? So those are called as what? Shine-Delgado sequences. These are situated where just 8 bases before start codon. Fine. So here, for example, say here ST sequences are situated. Here ST sequences, that is Shine-Delgado sequences, are situated. Now ribosome will randomly come and bind on this mRNA. Say, for example, it will bind over here. That means ST sequences will, you can say, provide a signal that now you have to start protein synthesis. So ribosome get activated. Now I have to do this work. I have to prepare now proteins. Right. So in case of prokaryotes, we have Shine-Delgado sequences. In case of eukaryotes, process is little bit Shine-Delgado sequences are not there, but certain POSUK sequences are there that we are not going to discuss in detail because that you don't have it in your syllabus, right? So POSUK sequences in case of eukaryotic organisms are present, which are just nearer to start codon only. Say, for example, here only. But in prokaryotes, they are situated 8 base pairs before start codon. Fine. Now ribosome will come over here, it will recognize and it will start moving in forward direction. Fine. So now for translation process, what is needed? What all things are needed for translation to get started? So we need what? We need mRNA. We need mRNA. We need ribosomes. We need ribosomes. Correct? In case of prokaryotic organisms, 70S ribosome is needed, whereas in case of eukaryotic organism, 80S ribosome is needed. Correct? You know now that smaller and larger subunit are present. In case of prokaryotes, 50S is the larger unit, 30S is smaller subunit. In case of eukaryotes, it is 60S and 40S ribosomal subunit. This we have already studied in detail before only. So just now focus that in prokaryotes it is 70S, in case of eukaryotes it is 80S ribosome. So mRNA, ribosome, and we need amino acids. Important thing, we need amino acids because proteins are made up of nothing but what? Amino acids. So who will carry these amino acids? Amino acids are not going to come by itself. Someone should be there to guide them. Now, which amino acid will come? Because how many amino acids we have? We have only 20 amino acids. Correct? We have only 20 amino acid, which code for how many codons? Which codes for 61 codons. 64 total 64 codons are there. Out of 64, 3 are stop codon. That means total 61 codons are there. So 20 amino acids are there to code for 61 codons. Now t-RNA is needed. t-RNA will be there to bring those particular type of amino acid depending upon the codons which are present on the mRNA. So we need what? t-RNA, that is transfer RNA. So we need transfer RNA. These are the requirements for translation process. Apart from this, we need certain factors. So we need certain factors to start and stop the process as well as to elongate the process. That I'll discuss in steps, right? Different steps. So what are these steps for translation process? First step is charging of tRNA. First step is charging of tRNA. Now, what is charging of tRNA? You know that the tRNA, we have studied two structures of tRNA. One is clover leaf model, second one is hairpin loop model. So in translation process, clover leaf model will become little bit difficult to draw each and every time. So we will draw this hairpin model of tRNA, right? So 5 prime is the start position, 3 prime is the end position where three bases are free, that is C, C, A. This one is the 3 prime position, also called as acceptor arm, whereas this position is called as anticodon position. This is anticodon loop, whereas this loop is called as acceptor loop or acceptor arm. Acceptor arm which will accept the amino acid depending upon the type of anticodon which is present, right? So what is charging of tRNA? Now this tRNA will be accepting a particular type of amino acid. Depend how I said told you that depending upon anticodon, this tRNA will bring particular type of amino acid, right? So amino acid will get attached over here with the help of energy. Of course, energy is needed. So that's why we are seeing charging of tRNA, right? Because energy is needed for that purpose. This charging of tRNA, that means addition of amino acids at 3 prime position of tRNA, right? So this is called as amino acyl tRNA. The charged tRNA, when amino acid get attached on the 3 prime end of the tRNA, that tRNA is called as what? Amino acyl tRNA. This process happens with the help of course, enzyme. With the help of enzyme, amino acyl tRNA, amino acyl tRNA synthetase. So this enzyme will be used for the formation of amino acyl tRNA. Right. So we have now this tRNA having this amino acid. Which one? This one, right? According to anticodon, since anticodon position will keep on varying. Say for example, this position is CCA, right? Say for example, it is CAA. So according to the anticodon, which is opposite of codon, amino acid will get attached over here. So so many types of codons are there, like 61 codons are there, and same codon may get repeated several times in one particular type of mRNA strand. So simultaneously amino acid tRNA will keep on forming. They won't wait that now there is requirement, now the process will happen. It won't be like that. There is continuous formation of this amino acid tRNA. When their role come, they will come and you can say help in formation of proteins. Correct? So this is called as charging of tRNA, that is formation of amino acid tRNA with the help of enzyme amino acid tRNA synthetase. That means addition of amino acid at the 3 prime end of the tRNA. Second step is, what is second step? Formation of proteins, the main step. Formation of proteins. Now this protein formation is now divided into 3 steps. Fine. What are those 3 steps? First step is initiation. Second step is elongation. Initiation, elongation, And third step is termination. Initiation to start the process. Elongation to, you can say long chain of proteins will be formed. Termination is to stop the process. Fine. Now, for initiation process, what we need? Process is getting started, right? So for initiation, we need this mRNA, we need a ribosome, we need tRNA, right? To come and attach, not only tRNA, we need amino acid tRNA, the charged tRNA. Fine. Along with this, we need certain factors. Remember, at the beginning, I wrote that we need certain factors. So those factors are called as initiation factor. In short, we can call it as IF factor. Right? So this initiation factor can be written as IF factor. So for initiation, we need three factors: IF1, IF2, and IF3. So these are nothing but what? Initiation factors. Similarly, for elongation process, we need elongation factors. Elongation factors. No specific name for those factors. So we'll be writing EF factors. EF is nothing but elongation factor only. For termination, we need termination factors. So this is termination factors, which can be indicated by TF. Okay. Now, how this process will get started? Let's start with these three steps only. First is initiation, the second is elongation, third is termination. But before that, let me tell you about ribosomal sites. We have two subunits of ribosomes, correct? This is larger subunit, this is smaller subunit. Now what will happen? See, we are having mRNA. So on this mRNA, this ribosome is coming and attaching. First, smaller subunit will come and bind. Then later on, larger subunit will come and bind on the mRNA. This ribosome will be having three sites called as E, P, and A. E site is called as exit site. Correct? P site is called as peptidyl site. P site is called as peptidyl site. Whereas A site is called as amino acid site. A site is called as amino acid site. You can assume there are three chairs or three doors, right? So one tRNA will come and will be assembled on one door or one chair. Then other positions will be vacant. Till the time those positions are vacant, another tRNA will come and sit on the another chair, right? So try to understand this process. See, now, since we know this mRNA, say for example, it got start sequences here only. Say for example, these are the start sequences only where ribosome got attached. So first tRNA will always come and bind, since all three chairs are vacant, right? All three chairs are vacant. So first tRNA will come and bind on this P site, peptidyl site. First tRNA will always come and bind on the P site. Now, since P site is already occupied, right? So the next tRNA which will come, it will come and bind on the A site. Okay. Since P site is already occupied, so second tRNA will come and bind on the A site. Now this tRNA first which came on the P site having this amino acid, right? Second tRNA will come and bind over here having this second amino acid. So what will happen here? There will be formation of bond between these two amino acids. There will be peptide bond formation between two amino acids. And this first tRNA will transfer its amino acid to the second tRNA. I'll try to draw only tRNA now. See, this was the first tRNA having this amino acid. This is the second tRNA having second amino acid, right? They are attached on the mRNA. This is present on P site. This one is present on A site of the ribosomes. Remember, sites are present in ribosome only. So there is formation of peptide bond in between these two amino acids. By that time, what will happen? This tRNA, this tRNA, there will be peptide bond formation, as well as this amino acid will get detached from this tRNA. So now what will happen? See, here, sorry, here it got detached from this tRNA. So what will happen now? See, ribosome will always keep on moving on this mRNA. Ribosome is going forward, correct? So mRNA is present on one position only. Ribosome came, bind, and it will keep on moving in forward direction. RNA is not moving. Ribosome will keep on moving. So what will happen? The sites which are present on this ribosome, they will also move, along with you can say ribosomal unit. So what will happen? This tRNA will take an exit. The work got over. tRNA came, brought amino acid, gave it to another tRNA. Now this tRNA will take an exit. From where? From this E site of the ribosome. Right. Now what will happen? See, since ribosome will keep on moving, how it is moving, and why they are called as peptidyl and amino acyl site. You got clear with the exit site, right? That means from the E site, a tRNA will take an exit. Why P site is called as peptidyl site? Because here at this position there is formation of peptide bond. That means this this tRNA which is present on P site is transferring its amino acid to the newly come tRNA, and there is formation of peptide bond. Right, so that's why this P site is called as peptidyl site. Why A site is called as amino acyl site? Because every time now new tRNA is coming on the A site. Only first tRNA came and bound on this P site. Every time new tRNA is coming and getting attached on the A site of the ribosome. So that's why. See, who is coming and attaching? Amino acyl tRNA. That's why the site is called as amino acyl site. Got it clear? Now, how this ribosome is moving? See, I'll draw another diagram. This is our ribosome. Okay. Now on this, sorry, mRNA. I'm so sorry. Now here we have this ribosomal units. Just a minute. So we have this ribosomal unit, right? Both the subunits joined together, and we have this EPA site. Now what will happen? This ribosome is already smaller subunit first came and was present on the Shine-Dalgaro sequences. It formed bond with the Shine-Dalgaro sequences. Recognized that the start codon is about to come. Say for example, start codon has arrived, reached. This ribosome got you can say assembled on that start codon. Now what will happen? This ribosome will start moving in this forward direction. It will keep on moving and keep on reading the sequences present on this mRNA. Fine. Now, what will happen with the tRNA? Suppose, for example, here we have AUG, right? We have start codon AUG over here. Similarly, other sequences will be present like this. Say for example, here stop codon UGA is there. This is 3' end. This one is 5' end. Now, tRNA came. This one is our first tRNA, which came on AUG side. So this is AUG having this amino acid. You can guess which amino acid will be present since it is AUG, right? So which amino acid? It will be methionine. So I'm writing in short, that is MET. Everything we are not supposed to remember that which amino acid is coming. For that we need to remember each and every codon, which codon is coding for which amino acid. So we will in short write amino acid 1. AA1 means what? Amino acid 1, because first amino acid is coming. Fine. Now second amino acid, say for example, I'm drawing here, it came having second amino acid, right? So I'm writing it as AA2, came and bound on this position. Now what will happen with the ribosome? Remember, ribosome is already attached, and I have just separated it to show you clearly that how it is working. See, this was the position of the ribosome. Now first ribosome, here ribosome is present over here, P site was vacant. So first tRNA came and bound on this P side carrying first amino acid. Second tRNA came on the A side. Correct? Since this chair was vacant, first P chair was occupied. Now A chair is vacant. So this tRNA came and bound on the A side. So here there will be formation of peptide bond. Fine. Peptide bond will be formed. Now amino acid got transferred. First tRNA work got completed. Now it has to take an exit. But how? Right? This ribosome now start moving in the forward direction because they need space, amino cell space for the next tRNA to come. So they need to vacate that chair. So how vacating is, how that vacant position will be available now? This ribosome will move in this direction. So see now how this ribosome will move like this. Ribosome is moving. See, ribosome is moving. Now first tRNA came on which side? E side. First tRNA, since ribosome got little bit moved in forward direction. So first tRNA came on the E side, right? Remember here there was formation of peptide bond and this amino acid work got completed. Now this tRNA came on which position? E position. Now which chair is vacant? A chair is vacant. Now every time E and P will be occupied, ribosome will move further. Every time A position will got vacant by tRNA, then new tRNA will come on the A side. That means every time ribosome is moving forward, the position of the tRNA is getting shifted, right? So the tRNA which was present on the P side got shifted to the E side. Now, since it is present on the E side, this tRNA will take an exit. That's why we are saying this side is exit side. Now, A site is vacant. Ribosome move further, right? Now, what will happen? Just give me a minute because I need to show that ribosome is moving, no? So, wait. Now, this tRNA will take an exit. Remember the position which I have drew. Now again, third tRNA is coming, having this was amino acid 2. This is amino acid 3, having third amino acid. Now, again, since there was formation of bond in between these two amino acid, this tRNA got free. Now here also similar process. What? That here there will be peptide bond formation between two amino acid. The second tRNA also got free. This tRNA also got free. Correct? How many amino acids now do we have? We have 3 amino acid chain. That means every time new tRNA which is coming is getting the amino acid which have already present. So all the previously present amino acids are added on the newly coming tRNA, right? Now again, we will come back to this ribosome. See, where was this ribosome initially? Here, right? So it was moving. Now this first tRNA came on which position? E position. So this tRNA took an exit. Fine. Now new tRNA came on the third position on the A side. Now this ribosome will again move in this direction like this. Again, the second tRNA now reached the exit position. Right. Now again, P site is occupied, A site is vacant. So this A site is now vacant. Since this A site is vacant, now again new tRNA will come on this A site. Every time there will be new tRNA which will come on the A site. So this will be amino acid 4. That means these codons are read properly by these tRNAs, right? So these are the codons which are read by the ribosome. tRNAs are coming. fourth amino acid came, there will be formation of again peptide bond over here, right? So this ribosome will take and move in the forward direction. Now this ribosome will again move like this. Just a minute. So yes, this ribosome now start moving. As and when it is moving, every tRNA is taking an exit from the E side, right? So now, since we are saying that fourth tRNA came, so fourth tRNA came on which position? A position it already came. So I'll just draw that same thing. A position, currently A position is occupied. Now ribosome will move forward. Now this process will keep on continuing. Now what we are observing is that whichever tRNA is present on initial site, like we have EPA site. Initially tRNA was present on P site. Every time P site tRNA will get shifted to E site, A site tRNA will get shifted to P site. There is shifting of tRNA from A to P, from P to A. And due to this process, this position will always remain vacant and the new tRNA will come and bind on the A site. So there will be formation of chain of these amino acid. That chain of amino acid depends upon how many codons are present over here. That much long chain of amino acid will be formed. Say for example now, this ribosome have reached over here, right? So this ribosome have reached over here having EPA site. At last position, this will be having, say for example here, it will be having tRNA, having long chain of amino acids. I repeat that the number of amino acids depends upon the codons which are present on the present on the mRNA. Right. So here last tRNA will come and bring this amino acid. Here, of course, tRNA was there, which donated this whole chain of amino acid to the newly coming tRNA, and it will take an exit. So every time from here there was in tRNA which was incoming. From here there was an exit of the tRNA. So since now this is last position here, last amino acid will get combined with the you can say the previously present tRNA, and this chain will got completed. Now how this process is getting completed? See, initially process got started by using smaller subunit and larger subunit of ribosome, right? For that Shine-Dalgarro sequences were used and tRNA came and bound. So for that initiation factors were needed. So now for elongation process, as and when this ribosome was moving, elongation factors were needed. So we wrote that only, that in during in steps only, that elongation factors are needed. Now the process is about to complete. How? Because at last this stop codon is present. Now UGA does not code for any amino acid, right? So when this ribosome will reach on this stop codon, there won't be any tRNA having amino acid, because this stop codon does not code for any amino acid, right? So since this is the signal for this amino acid, there should not be gap, that it won't move further, like stop codon is there, none of the amino acid is coming, okay, I'll move further, I'll receive another amino acid. There should not be gap, right? So there should not be a breakage of the link for the formation of protein. So stop codon is coming, no amino acid is coming, immediately the termination factors will come. They will release this you can say ribosomal subunit, these tRNA will get released, and this finally formed chain will get released. So here finally what is happening, here termination is happening. So for termination factors will be needed. For termination, after termination, we have this chain of amino acids. So termination factors will help in releasing of each and every part of this translation process. So this chain of amino acid is nothing but a protein. It will act as what? Protein. I have drawn few amino acids only, right? But number of amino acid will be too much large because proteins are complex structure in nature. The structure can be little bit complex to more complex. It all depends what type of protein is there, what function they are having. So long chain of amino acid will be formed. So with the help of termination factor, this amino acid chain got released, right? All these tRNA will got released. This smaller subunit and larger subunit of ribosome got released. And simultaneously, this mRNA also got free in the cytoplasm. So with the help of termination factor, everything got released and we got this protein. Fine. So certain important things we need to remember that for initiation of this process, always first smaller subunit will come. One more important thing is remember that for the starting of the process, initiation sequences was AUG. Correct? So for AUG, this tRNA came. This tRNA was having anticodon sequences. Anticodons are opposite of codon, correct? So AUG, opposite of AUG will be UAC. That means UAC sequences are present on the tRNA. So this tRNA brought this methionine amino acid, correct? So in case of prokaryote, I'm talking about this tRNA, first tRNA always, right? In case of prokaryotes, first amino acid, first amino acid that is methionine is always formylated in nature. What do you mean by formylated? Formyl group is added on the methionine amino acid so as to provide a little bit more stability, since we know that in prokaryotes coupled process is there. So in case of prokaryote, this is important. In case of prokaryote, first amino acid, that is methionine, is always formylated. So in short, we can write it as FMED. F-methionine, that means formylated methionine. Remember, I said only first amino acid, first methionine, right? Whereas in eukaryotes, in case of eukaryotes, methionine is normal methionine only. Methionine is normal one only. That means we won't be writing F-methionine in case of eukaryotes. Simply we will be writing methionine, right? But see, for example, in case of prokaryotes organism, we are saying first methionine is F-methionine. So, say for example here AUG was there. Can we say that AUG will come only once in whole mRNA sequences? No. It will keep on repeating, right? Initially, ribosome got AUG over here. That's why process got started. Now, stop sequences are here. Say, for example, here, UGA. So, in between also, AUG may come, no? Once or twice or thrice or many times, I don't know. How many times they are coming? We can't say anything, right? So, if they are coming, that methionine will be normal only, right? Here, only first methionine will be formylated methionine because at the start of the protein, you can say structure. So, in case of prokaryotes, first methionine will be F-methionine. Other methionine, if they are coming in the structure, those will be normal methionine only. Whereas in eukaryotes, all methionines will be normal NET only. Correct? So, this is what translation process is. So I'll just revise it a little bit in short. We have this mRNA, right? 5' to 3'. AUG sequence, say for example, it is present over here. Stop codon, say for example, UAA is present over here. The sequences which are present before and after the start and stop codon, those are called as untranslated region, UTR, right? In case of prokaryotic organism, just before this start codon, 8 bases before the start codon, SD sequences are present, which are nothing but what? Chandel-Garro sequences, right? This will be the start signal. This will be the start signal for translation process. Correct? For the process to get started, the ribosomal small unit will come and bind over here. Small unit of ribosome will come and bind over here. Then larger subunit will come, right? But as it is moving in forward direction, it will come and reach on the start sequence of this mRNA. So, say for example, here now we have this smaller subunit, ribosome got moved, smaller and larger subunit. Every time tRNA will come and bind on this start sequences or the other sequences when it is moving forward, sequences will be read by this ribosome. Every time new tRNA will come and bind on the codons by carrying this amino acid. First tRNA will always come on the P side of the ribosome. Every time now, next tRNA, whenever it will come, next tRNA will always come on the A side of this ribosome, and there will be formation of peptide bond between the first and second amino acid. Now this ribosome will move in forward direction. Since it is moving, tRNAs are present on the same position, but ribosome is moving. So the chairs on which those tRNAs are present, those will get shifted. That means P side tRNA will shift to the E side. A side tRNA will get shifted to the E side. Right? This process will keep on continuing till the stop codon is reached. So every time EPA, remember, first tRNA will come on P side. After that, every tRNA which is coming, it will come on the A side, amino acid side. There will be formation of peptide bond. These sites along with ribosome, they are moving. So that's why these sites will also get shifted like this. These are the ribosomal sites, right? So these ribosomal sites will get shifted. Now the tRNA, P site tRNA got shifted to E site. A site tRNA got shifted to P site. A site is vacant, right? Now new tRNA will come and bind on the A site, carrying new amino acid, right? It will help in the formation of peptide bond. By that time, this tRNA will get detached, it will get released from this amino acid and it will take an exit. Now again, same process that what this ribosome is getting shifted, this ribosome is getting shifted. So again, A site is vacant, tRNA will come and bind on this A site, and similarly, the process of protein formation will continue. Initiation is happening with the help of initiation factor. Elongation is happening with the help of elongation factor. Finally, releasing is happening with the help of release factor. So that is what translation process is. At the end, we got a long chain of amino acids. This long chain of amino acids, nothing but will be a protein. We got this protein by this process, right? Now, one question that as and when this ribosome is moving in forward direction, right? Say, for example, again, I'll draw one more diagram. This is my tRNA, mRNA, I'm so sorry. Here my ribosome is attached. There can we say only one ribosome is present? No, many ribosomes are present. And since this ribosome has moved from this position to this position, now this mRNA is vacant, right? Another ribosome may come and bind on this you can say on this mRNA, right? Another ribosome will come and bind on this mRNA. So there will be again second ribosome is continuing with the protein formation process. Likewise, third ribosome can also come. Now this ribosome has reached this position, right? And what will happen? The third ribosome will come. The third ribosome will come and bind on this position, right? Third ribosome came and was present over here. So simultaneously we can see many ribosomes can be attached on the single mRNA. That means same type of protein can be formed simultaneously. Many copies of the same protein can be formed when there is availability of many ribosomes. So this condition is called as polyribosomes or also called as polysomes. Okay, so remember this process, how process is happening. Process is very easy. Just you need to imagine that ribosome is moving. Ribosome is having three positions, EPA. Every position will be always occupied by one or other tRNA. If one is moving, another will come and sit on that position till the stop codon is coming. Right? So we need certain factors. Those factors are called as what? Initiation factor, elongation factor, termination factor. This is how this translation process is happening. I hope this topic is clear to all of you, right? So here we stop with this topic. I'll be coming again with a new topic, with a new concept. We'll be explaining different topics, right? Till then, bye-bye. See you.